Meiosis is a process of forming gametes or sex cells. Gametes have half of a full set of chromosomes and are known as haploid cells. In males, these gametes are the sperm, and in females, these gametes are the eggs. During interphase, much like mitosis, the cell replicates its chromosomes, the centrosomes split into two, and the chromosomes are uncondensed and have no shape yet. The cell to be split into gametes is known as a germ cell. Prophase 1 is the longest phase of meiosis. The nucleoli begins to disperse and break apart. During early prophase 1, each chromosome pairs with its homologue, aligned gene by gene, and crossing, otherwise known as a synapsis, occurs where the DNA molecules of non-sister chromatids are broken by proteins and are joined to each other. The homologous chromosomes are loosely paired, and each homologous pair has one or more X-shaped regions called chiasmata, where crossovers have occurred. Later in prophase 1, spindle microtubules from one pole or the other will attach to the two kinetochores, one at the centromere of each homologue. The two kinetochores of each homologue act as a single kinetochore. When the homologues move, it begins to form the metaphase plate. In metaphase 1, the chromosomes move towards the center of the cell in homologous pairs, which is also known as tetrads, forming a metaphase plate. The microtubules connect to the kinetochores of the chromosomes. Gregor Mendel had two laws. The first law was the law of independent assortment, in which alleles of genes of non-homologous chromosomes will assort themselves independently during gamete formation. The second law of segregation, states that two alleles from each gene will separate during gamete formation. In anaphase 1, the homologues move towards opposite poles, guided by the spindle apparatus. Sister chromatid cohesion persists at the centromere, causing chromatids to move as a unit towards the opposite ends of the cell. The breakdown of proteins that are responsible for sister chromatid cohesion along chromatid arms allows homologues to separate. Each of the new daughter cells are haploid and only have half of the DNA of a full double set or single set of DNA. Cytokinesis, or the splitting of the cytoplasm, happens simultaneously with telophase 1, while in plants the process involves the cell plate, which forms into the cell wall. In animal cells, it involves a cleavage furrow, which is a contractile ring that pinches the cell into two. No further DNA replication occurs. During prophase 2, a spindle apparatus reforms after the centrosome replicates again. In prophase 2, chromosomes, each still composed of two chromatids associated at the centromere, move towards the metaphase 2 plate. During metaphase 2, the chromosomes are positioned at the metaphase plate as in mitosis. Because of the crossing over in meiosis 1, the two sister chromatids of each chromosome are not genetically identical. The kinetochores of sister chromatids are attached to microtubules extending from opposite poles. During anaphase 2, the breakdown of proteins holding the sister chromatids together at the centromere allows the chromatids to separate. The chromatids move towards opposite poles as individual chromosomes. In telophase 2, the two daughter nuclei form and the chromosomes begin decondensing. Cytokinesis occurs again, involving a cell plate in plants and cleavage furrow in animals. The resulting four cells are genetically distinct haploid daughter cells. In males, the four gametes are sperm cells. In females, 3 of the 4 haploid cell products of meiosis become nurse cells and provide nutrients for 1 of the 4 egg cells.